So good afternoon. I'm Mary Howe Grant. I'm past president of the Science and Engineering Council of Santa Barbara, and it's my pleasure to welcome you to today's meeting, the August meeting. We are still on Zoom, obviously. We're talking about the logistics of moving to in-person meetings, but we haven't gotten there yet. But it's under consideration. So I'd like to introduce some other board members that are with us today. Bob Lilly, Yvonne DeGraw, and is Bill Theringer here? He may be. Okay. You all know by now, I think, that Community West Bank, Minot Enterprises, Bengal and Toyon are our sponsors, and we thank them very much for that. And we also thank all of you who have donated to the Science and Engineering Council, because those funds help us to cover our administrative expenses. I would like you to note that in addition to our luncheon meetings that we give monthly, we do science fair awards. Many of you attended that, or you may have attended the science fair meeting that we held this year. And we also give out scholarships via the Santa Barbara Scholarship Foundation, SEC scholarships. We have 2 recipients this year, we're very pleased to say, and we hope to hear from them personally later in the year. So thank you to all those who donated to and to support that scholarship, ensuring that there will be future scientists and engineers. Our scholarships are given in the STEM area. And with that, I guess I would like to turn the meeting over to our program chair, Yvonne DeGraw, and she can tell you what's happening with us for today and some of our plans for the future.
Great, thanks, Mary. Hello, everyone. Our next meeting will be September 14th, also at noon on Wednesday, and we'll be sending out an email soon with information about the topic for that meeting and the speaker. Today, we're happy to have Dr. Chris Richardson as our speaker. He's an assistant professor at UCSB in the Department of Molecular, Cellular and Developmental Biology. He joined the UCSB faculty in 2019. Dr. Richardson received his BS in bioengineering from UC San Diego and his PhD at UC San Francisco. His postdoctoral research at UC Berkeley investigated DNA repair in mammalian cells and how DNA repair processes can be used for gene editing. He's also worked for Spotlight Therapeutics, a biotechnology company in the San Francisco Bay Area. He's speaking today to us on using gene editing tools to understand DNA repair. While you're watching the talk, you can see subtitles by enabling live transcript in the Zoom application. And you can ask questions today in the chat box or after the talk by unmuting yourself. So Dr. Richardson, we're happy to have you here.
Happy to be here. Thank you very much for the invitation and for kind introduction. Can everyone see my slides?
Yes, thanks.
Okay, great. So I put a lot of introduction into the beginning of this talk, and then towards the end, I want to talk about 2 stories that we're working on in my lab right now. So big purpose of my lab is understanding how cells fix DNA. And so what, in this case, are we talking about when we're talking about DNA? So DNA is nucleic acid. It's in the nucleus of just about every cell in your body. And so DNA itself is a double helical molecule. That's what's shown on the right side of this slide. And there's a whole lot of it in the nucleus. So the nucleus of human cells is about 5 microns in diameter. And there are about 6 billion bases of DNA that are packaged into that nucleus. And so the DNA is condensed into these filamentary structures, and those filamentary structures are condensed into the big X-shaped chromosome, and then the individual chromosomes are packaged inside the nucleus. And so the packaging is fairly intense. If you treat the cells chemically in such a way that you undo this packaging, the cells just explode. DNA, comes out like spaghetti. So stored in the nucleus. It's highly packaged and regulated, and it's important because it encodes the instructions that the cells use to make RNA and protein molecules that they use to do sort of biologically interesting work. So DNA is often described as the blueprint of the cell. And so DNA is not just static, it's constantly under threat, both from environmental factors and from endogenous factors. So DNA is constantly being damaged. Those environmental factors can be things that we all know are bad for us, ionizing radiation, various chemicals, but actually these are fairly rare sources of DNA damage. The majority of the DNA damage comes from endogenous sources. So replication, when DNA is replicated in preparation for a cell to divide into two cells, when DNA is transcribed to convert that information encoded in DNA into RNA, and even the process of cellular metabolism, all of these things can cause large amounts of DNA damage. So our cells use oxygen to produce energy. In doing so, it can produce various reactive chemicals which can damage DNA. And so the idea that I want you to keep in mind is that cells don't really get a break from this. It's not like there's a pulse of DNA damage and then there's sort of a period of repair. There's constantly damage being created and the cells are constantly having to fix this. And the consequences of not fixing it can affect a specific cell. So if the DNA is damaged, if it's not repaired, or if it's not repaired correctly, it can change the sequence of the DNA. So we call this mutations. If it's severe enough, it can also lead to cell death. These can also cause, DNA damage can also cause organism-wide problems. So sort of one of the really interesting areas of aging research at the moment is this idea that DNA damage can sort of create this chronic level of stress, which maybe plays a role in the development of age-related diseases or autoimmune disorders. As you might imagine, there are many different types of DNA damage that can happen. So DNA is a double-stranded molecule. One of the strands can be severed to produce single-strand breaks. Both of the strands can be severed to produce double-strand breaks. You can create crosslinks within a single strand of DNA. You can add bulky chemical groups that covalently modify DNA. You can covalently link the two strands of DNA together so that they can't be separated, or you can create mismatches where the sequence on one strand of DNA doesn't match the sequence of the other strand. And cells have to be able to repair an awful lot of these. So these are very rough estimates, and this is all per cell per day. So for cells growing in a dish, each cell might experience 1,000 single-strand breaks per day, 40 double-strand breaks per day, 100,000 intra-strand crosslinks, 100,000 DNA adducts, or 100 inter-strand crosslinks. And so again, it's this continuous process of just fixing this damage that gets introduced. And so my lab is primarily interested in one type of damage, double-strand breaks. So I'm interested in these for a couple of reasons. First, they're incredibly toxic to cells. So there's this line of research that suggests that even a single double-strand break that's unrepaired can be lethal. And so one of the themes that will come up later is that double-strand breaks have to be repaired. Cells have to fix them. And it's not enough to just fix them, they have to be repaired correctly, right? So in a double strand break, you have two loose ends of DNA, and what you want to do is stick them back together. If you stick those loose ends somewhere else in the genome, it can cause all manner of problems. You can have, the general term for it is genome instability, but you can stick two genes together that are not supposed to go together and create new and potentially dangerous proteins. You can create structures that don't allow cells to divide. Misrepairing these things can have profound consequences for the cell. They're caused by ionizing radiation. They're caused by a variety of chemicals. In fact, a lot of chemotherapeutic agents that are used for cancer treatment cause double-strand breaks. And they're also caused by normal cell biology, mostly processes like replication. And so again, they're extremely toxic to cells. And so if there's a double strand break, the cell needs to repair it. And so I kind of wanted to try to explain why I think double strand break repair is difficult. And I'm going to use a crude analogy in order to do that. So, if you come across a broken rope, you as a person will go through a series of steps to try to fix that issue, right? And your workflow may differ, but this is the one that I came up with. So, the first step is identifying the problem. It's easy to see this rope is broken, right? And then, using what I know about ropes, I can propose a solution, right? I might tie it back together. If it was made of plastic, I might melt it back together. I could stitch it back together. I could go so far as to tie each individual strand back together. There are a lot of different approaches that I could take. And then I would want to test that solution. So, tug on it, see if it would support some weight, maybe even get a new piece of rope if I needed to, and then return to, whatever normal use this piece of rope had, hanging a hammock or whatever. But cells can't really do any of this stuff, right? They can't see a double strand break. They don't see the world in the same way that we do. And because they can't see it, problems like how do you know that both ends of the double strand break are still in proximity are much more relevant for the cells fixing these kinds of lesions than for us fixing a rope that's been cut in two. Another major challenge that the cell faces is, DNA is continually in use. These cells are continually replicating their DNA. They're continuing to transcribe genes. And so they don't really have an opportunity to stop their cell biology for long periods of time and implement some kind of fix. And there's another question, which is not so important for this talk, but an important topic in the field, and that is, if the cell identifies DNA damage and fixes that DNA damage, should it trust the region of the genome that it has fixed, or should it somehow remember that the status of that has changed? And so these are all challenges that the cell has to overcome in order to fix double strand breaks. So how do cells fix double strand breaks? They have a lot of different pathways that they can use, and when we say pathways, generally what we mean is you can separate them. They rely on the function of different repair factors. But these different pathways tend to cluster into two areas. The first is known as end joining. And so this is ligation based, and conceptually it's very simple. It's just the cell is just going to stick the two ends of the double strand break back together. It's relatively fast. It can take place in minutes, and it's a little bit risky because the cell doesn't actually know, it can't see the double strand break. It doesn't know what the sequence is supposed to be. And so if in the time that it takes to stick this back together, one of the bases at the end of the double strand break gets clipped off, or maybe one of them gets extended, it can stick the molecule back together inappropriately and create these things called indels, which are insertions or deletions. They're just a type of mutation that can disrupt the function of the underlying region of the genome. So that's end joining. The second pathway is known as homologous recombination. And before I talk about that, there's one important term that I need to define, and that is homology. So when biologists talk about homologous sequences, what we mean are sequences that have the same identity. So if it reads ATGC on one strand, it reads ATGC on the other strand. So homologous recombination is recombination between identical molecules. And conceptually, what that means is the cell takes the double strand break and it repairs it using a third molecule that has identical sequence to the broken molecule. And we'll get into the mechanism of this in the next slide, but this is a really complicated process that involves the activity of dozens of DNA repair factors. And as a result, it's slow. So we start to see homologous recombination finish after about 24 hours. So much, much slower than end joining. Homologous recombination has two really interesting properties. First of all, because the cell is using a template, this third molecule to fix the double strand break, it means it can kind of check its work. And so it has the potential to result in very high fidelity repair, low probability of mutations. We as scientists, as gene editors, can also use the process of homologous recombination to insert new genes. So if we take this template molecule that's shown in green here, and we add a new gene into the middle of it, we can often, if we get the parameters right, get that new gene to insert into the site of the double strand break. So two different groups of repair pathways to fix a double strand break and different benefits and drawbacks from the different pathways. So I thought I'd just briefly cover the mechanism of homologous recombination. So the two molecules shown in step one here, there's a double-stranded molecule in gray that contains a double-strand break, and there's a double-stranded molecule in blue, which has identical sequence to the molecule in gray that's intact. So the top molecule has the double-strand break, the bottom molecule is the template. So in step two, what's going to happen is the molecule with the double strand break is going to be resected. So the double-stranded DNA right at the break site is going to get chewed back to expose these single-stranded DNA tails. And then in step three, those single-stranded DNA tails are going to invade from the molecule containing the double strand break into the molecule containing, the molecule is acting as a template. In step four, the single strands that have invaded into the template are going to begin synthesizing. So they're going to copy template sequence. And then eventually in step five, that copying process is finished. And if you trace the outline of the green molecule, you can see that it's now intact. So all of the sequence of the double strand break has been restored. And the cell just has to resolve the intermediate in step five. So if the cell goes through and just cuts the links holding the gray molecule and the blue molecule together, you'll go back to two double-stranded DNA molecules, and they should have identical sequence again. So there are a couple of things that I'd like you to keep in mind from this slide. The first is that homologous recombination is a complicated process. And the second is that every step that I just described and dozens of steps that I didn't describe involves the activity of a specific repair factor. And those repair factors have to act in a specific order over a very long period of time, right? So this is a really complicated and risky workflow that the cell needs to execute. And so that really sets up why I'm interested in this step that I've highlighted in the blue box here. So we think that the cells make a decision at this point. And it's not decisions in quotes because it's not a decision like we would make a decision, but it does have to commit either to end joining or homologous recombination. And this is a difficult decision to make in two ways, right? First of all, this is an irreversible step, and there's a big difference in the amount of time that it'll take to complete either of these different types of repair. You know, end joining is quite fast, but it has the potential side effect that it could cause mutation. Homologous recombination is quite slow and should be relatively error-free, but if one of those steps doesn't complete, it's probably going to kill the cell, right? Because it's extensively altered the DNA so much in order to perform this homologous recombination process. And so the second reason I think this is interesting is there are real stakes involved in it for the cell, right? It really has to weigh the likelihood of mutations caused by end joining being toxic versus the likelihood of homologous recombination failing being toxic. Okay, so why study this process at all? So there are three reasons. First of all, I think it's really important for basic biology. So double-strand breaks get caused by all kinds of things. They get caused by ionizing radiation. They get caused by chemotherapeutic drugs. They're even caused by endogenous biological processes like transcription or replication. And so understanding how cells respond to these kinds of stresses allows us to design new interventions, to sort of understand how different fields are linked together. There are really great biological reasons to try to understand this. It's also very important for gene editing in the biotech sense. So, you know, what's cartooned here is a fluorescent rabbit. That's not something that would be interesting from a biotech perspective, but there are many things that are produced today that involve taking genes from one organism and sticking them into another organism, right? And so that's a case where you would want to understand how double-strand break repair worked. And it's also and increasingly important for therapeutics. So these are treatments that are just starting to go into patients today. And I'll try to run through two very vague examples, and then I'll give you the specific genes in a minute. So if we consider two patients, patient A has a gene P, which confers a weak cancer immune response. So this patient's immune system is weakly interacts with a tumor that they have. Patient B, on the other hand, has a mutant form of gene H. And so this mutant form alters the shape of this patient's red blood cells and causes a number of other disease symptoms. So we would want to treat these patients in different ways. So for patient A, what we might do is introduce a double-strand break into gene P, and then rely primarily on end joining outcomes to disrupt the function of gene P. And so we take this patient from having gene P to having this mutant form gene P star, right? So if gene P confers a weak response to cancer, gene P star confers a strong immune response to cancer, right? So it's more likely that this patient's immune system will respond to the tumor that they have. For patient B, on the other hand, what we might want to do is introduce a double-strand break in gene H star, and also introduce DNA that encodes normal gene H into this patient, and then rely on the HR pathway to reverse gene H star to gene H. And so if the patient now has gene H, their blood cells have normal morphology, and many of the symptoms for this disease disappear. So two different patients, two different diseases, very different treatment protocols, but they do have some things in common. The first thing that they have in common is that both of them meet this definition that I introduced earlier on, where we've introduced double-strand breaks in those genes, and those double-strand breaks have to be repaired. And control over the decision, the cell's decision for how to repair those double-strand breaks could improve treatment outcomes for both of these patients, right? So in the case of patient A, we're relying on EJ outcomes to disrupt the function of a gene. And so we would want to sway that decision towards EJ in the case of patient A. In the case of patient B, we're trying to replace a mutant form of the gene. And so we would want to favor HR outcomes. So we want to sway the decision towards HR. And so this has been very vague, but there are clinical trials right now to do both of those things. So patient A, the actual treatment they're receiving would be inactivation of a gene called PD-1 in their T cells. And PD-1 kind of acts as a brake on the immune system. So if you take the brakes off, the immune system gets much more active. You get much more reaction between the immune system and the tumor. Patient B, on the other hand, has sickle cell anemia. And so if you reverse the mutation that causes sickle cell anemia, then it restores normal morphology to the blood and you lose a lot of the other symptoms. And so another thing that these patients have in common is these are both gene editing treatments that are used on blood. And so for safety and efficacy reasons, if you were treating these patients, you would harvest their blood cells, you would perform these gene editing operations on their blood cells, you would check that everything was what you expected it to be, and then you would reintroduce blood into the patient, right? So you're not systemically dosing with editing reagents or anything like that. So as I mentioned in the previous few slides, these workflows all start with introducing double-strand breaks at targeted regions in the genome. And this is something that historically was quite hard to do, but became much easier about 10 years ago with the discovery and subsequent popularization of this molecule called Cas9 by Jennifer Doudna and other scientists. So what Cas9 is it's a bacterially derived protein, and it has two very important activities. The first is that it can cut double-stranded DNA. So it's a double-stranded DNA endonuclease. And the second important thing that it can do is it can be almost infinitely retargeted to different regions within a genome, a human genome, a plant genome, whatever. And it does so because Cas9 holds tightly onto this guide RNA molecule. And the region of the guide RNA shown in green can have the same sequence as the region of the genome that you want to introduce a double-strand break into. And so if the guide RNA sequence is the same as a region of the genome, Cas9 is going to find and bind that region of the genome that matches the guide RNA sequence, and it's going to introduce a double-strand break there. And so by varying the sequence of the guide RNA to target gene P or to target gene H, you can introduce double-strand breaks specifically where you would like. And so this is the thing that I started doing when I was a postdoc. So Cas9 is a gene editing reagent. It's used in the workflows that I talked about in the previous slide. But fundamentally, what it's great at doing is introducing targeted double-strand breaks at specific regions of a genome. And so if you know where you're introducing a double-strand break, if you know when you're introducing it, you can study its repair in great detail, which is the main focus of my lab at UCSB. So just to review the introduction. So nuclear DNA gets frequently damaged with double-strand breaks, and cells have multiple ways they can fix this damage. So end joining works by sticking breaks back together. Homologous recombination works by copying the missing sequence off of a third molecule, which is identical to the molecule that contains the double-strand break. How cells choose end joining or HR is a mystery. But understanding the details of that, so understanding how a cell chooses EJ or HR may improve either biotechnology or therapeutic applications. Give me 1 second. I can't advance my slide. Okay, there we go. All right, so in the lab, how do we study double-strand breaks? So we follow a 3-step workflow. So the first thing we do is induce double-strand breaks. And for the most part, we do that with targeted nucleases like Cas9 that I just talked about. And then in many cases, we allow the cells to fix the breaks and we visualize the repair outcomes. So we can do this very systematically. We can look at the sequence of the DNA surrounding where we know we introduced a break. And by looking at how that sequence has changed after the induction of the double-strand break, we can infer what repair processes were active. We can also take a shortcut and we can do something like introduce green fluorescent protein into the cells, which we know is only going to be introduced by the HR pathway. So if we induce double-strand breaks in our cells, wait 4 days, and look at the frequency of cells that express GFP, we know the rough fraction of cells that underwent HR as opposed to other forms of repair. And so this is our standard workflow. We've also started looking into this black box shown in step 2 here. And so what we can do is we can look at earlier time points during double-strand break repair itself, and we can look at repair factor localization. We can look and see if various repair factors are associating with DNA, and we can see sort of intermediate DNA changes that might occur. And so double-strand break repair is a very well studied topic. I think there are really two original perspectives that my lab has on it. The first is that we hypothesize that there is an intermediate step that arises before the decision is made. And this is an intermediate step where the cell can simultaneously explore either end joining or homologous recombination outcomes. So instead of having to sort of take the leap and assume that it's going to be able to finish however many steps it needs in order to do homologous recombination, maybe the cell can start doing many of those steps before it makes the decision. And so, committing to end joining involves removing these HR factors indicated by the yellow ovals, and committing to end joining involves removing the purple circles and committing to homologous recombination. And so that's one theory that's specific to my lab. The second theory that's specific to my lab is that these repair factors indicated by the yellow circles exist at all. So we think that there is a population of DNA repair factors that specifically acts on the template molecule. And to my knowledge, we're the only people that are kind of going after this. We know a lot about repair factors that act at the double-strand break. It's much harder to find factors that act specifically on the template. So I'm going to talk about 2 projects today. The first is run by graduate student Christine, who's interested in finding repair factors that are present on this intermediate structure. And the second is a project run by another graduate student, Hannah, who's very interested in looking for DNA repair factors that act specifically on template molecules. So the start for both of these projects was an observation that I made as a postdoc, that if you inactivate one specific DNA repair factor, FANCD2, you're no longer able to do homologous recombination, but you can still do end joining. So it's almost like FANCD2 is playing some role in allowing cells to do homologous recombination. Like many other things in this talk, the regulation of FANCD2 turns out to be very complicated. So very briefly, what happens is cells uncover lesions in step 1 and then activate a multi-factor complex called the FA core complex, which modifies FANCD2 and FANCI. So a heterodimer of FANCD2 and FANCI together. This modified version of FANCD2 localizes to the site of the lesion, and then somehow, we don't know how, that's the event that actually triggers the repair, in this case of the double strand break. And then once the lesion has been repaired, FANCD2 gets recycled back to its inactive form. And so really the key step, the key thing that my lab and other people in the field are trying to understand is, what is FANCD2 actually doing here? Like what is its actual purpose? And so, one of the early observations we made was that if you make cells that don't have FANCD2, they can't do homologous recombination. And so that could happen for a number of reasons. So a subsequent question we asked was, does FANCD2 physically associate with sites where we're introducing double strand breaks? And so we asked that question directly. We introduced double strand breaks using Cas9, again, targeting a region of the genome we know in advance. We then grab on to the FANCD2 molecules that are in the cell, and we pull them out, and we look at what the DNA sequences they're bound to are. And we look at how that overlaps with where we know we're introducing the double strand break with Cas9. And so the top profile shows that FANCD2 abundance is on the y-axis and chromosome position is on the x-axis. And so the point of maximal FANCD2 abundance overlaps with the point where we know we're introducing the double strand break. So FANCD2 is required for FANCD2 is required for homologous recombination, and FANCD2 binds directly to the double strand break. So it seems like it's really crucially involved in this process. But there is a challenge in understanding this data, and that challenge is that these cells are undergoing homologous recombination, and we know that the template sequence and the double strand break sequence are identical, right? So is FANCD2 actually associated with the double strand break molecule, or is it associated with the template molecule? And figuring that out turns out to be very difficult. So there are kind of 3 models that are possible, right? So FANCD2 could be bound only to the double strand break. It could be bound only to the template molecule, or it could be bound to some kind of repair intermediate that arises with conjunction between the double strand break and the template molecule. So the way we tried to answer this question first was we took another protein, MRE11, which we know binds only to the double strand break. And we just asked if FANCD2 binds to the same DNA as MRE11 or different DNA from MRE11. So if we induce a double strand break and we look at the localization of MRE11, MRE11 localizes to the double strand break just as we expect. If we do the same experiment with FANCD2, we see FANCD2 also localizes to the double strand break, and it does so with a much broader distribution. Now, if we do control experiments where we sequentially pull on FANCD2, we see the same distribution. It still associates with double strand breaks. But if we do a sequential experiment where we first pull, okay, so the conclusion from these experiments was that FANCD2 and MRE11 were bound to separate DNA molecules. And so the big question is, what are these separate DNA molecules? So in the cartoons that I've shown you of the templates so far, we've sort of treated them as these linear DNA molecules that are identical to the site of the double-strand break. And that is true, but there are also sequences outside of that, shown in gray here, that are not homologous to any region of the genome. So they don't match anything in the human genome. And so if you were to, for example, pull on Fanconi and see those DNA sequences associated with it, it would be very clear evidence that Fanconi was associated with template DNA and not double-strand break DNA. So if we do those experiments, that's exactly what we see. So Fanconi definitely binds to the template molecule in cells. And so, I think this really rules out the first of the three possibilities that I introduced, which is that Fanconi is only bound to the double-strand break, because we know that it binds to the template as well. So it could be that it's a template-specific molecule, or much more likely, Fanconi is actually bound to some repair intermediate that's being formed during DNA repair. And so, the follow-up experiments that we're trying to do at this point is are first of all to get better resolution on where Fanconi is bound. So one of the ideas that we've discussed is basically marking the template DNA in some way so that we can more easily see the difference between Fanconi bound to the double-strand break and Fanconi bound to template DNA. And we're also interested in getting more mechanistic insight. So when we talked about activation of Fanconi, it gets activated by ubiquitination. And so we'd like to know if the population of Fanconi that's bound to the template molecule or bound to repair intermediates is post-translationally modified with these ubiquitin molecules. Okay, so Fanconi binds to template DNA. And another question that we've been trying to answer, and actually the answer to this question is what ultimately brought me here to talk to all of you today, is are there other repair factors that bind specifically to templates? And so the general workflow that we followed to try to answer this question is we induce double-strand breaks in cells, and we provide those cells with template molecules that are that we can then pull back out of cells. So we can affinity purify those template molecules back out. And then we can do what's known as proteomics to identify any sort of DNA repair factors that are bound to those template molecules. And so we know that Fanconi binds template molecules during double-strand break repair. We want to identify other repair factors, other proteins that are also involved in this process. And so the strategy that we used to try to complete this workflow was to take our template molecules and then treat them with a molecule called sorrelin biotin to produce a structure that is shown in cartoon form on the right. So basically, we have template molecules that have this small molecule called biotin hanging off of them, and we can then grab onto that biotin and we can pull the whole template molecule back out of cells. And so the chemistry behind this is pretty interesting. It introduces into the template molecules another type of DNA damage, which I mentioned briefly at the start of this talk. It introduces inter-strand crosslinks. And so the really interesting thing that we found was when we made these biotinylated template molecules containing these interstrand crosslinks, we got much higher frequencies of homologous recombination. And this was instantly and immediately interesting to us because one of the perspectives we have on double-strand break repair is that things that happen on the template may bias end joining or HR outcomes. And here we have, great evidence of that. If we modify the template molecules to introduce this interstrand crosslink, it's now much, much more likely that the cell will use those template molecules to perform homologous recombination. And so we basically turned this into a tool. So we worked out many of the parameters in terms of how many crosslinks we could add and where we should add them. And then we started using them in workflows, either for regenerative medicine or immuno-oncology, where people want to introduce genes into specific cell types by homologous recombination. So we can insert genes into iPS cells. There's no current uses of these for regenerative medicine applications, but there's a lot of interest in it. And basically, we can the gray bars here are cross-linked templates, and the black bars are our uncross-linked templates. And so the cross-linked templates are much more efficiently used as substrates for homologous recombination, both in iPS cells or in patient T cells, which is shown on the right. The next thing we wanted to, so we think that this is a useful technique for boosting the frequency of homologous recombination in biotechnology and therapeutic applications. And the next question we wanted to know the answer to was whether or not which pathway all of this acted through. And so to answer this question, we drew up a list of all of the repair factors that fix interstrand crosslinks in cells, and we generated cells that are free of those factors. So our control population of cells is on the right. So our HR frequency with uncross-linked templates is shown in black, with cross-linked templates is shown in gray. And so in our control population, cross-linked templates are much more efficient than uncross-linked templates. And this is true if we eliminate factors in the base excision repair pathway, the BER pathway here. If we knock out factors in a transcription-coupled nucleotide excision repair, that's TC-NER pathway, the global genome nucleotide excision repair pathway, that's GG-NER, and also these NEAL3 factors that are involved in direct excision of crosslinks. But where we really see some interesting behavior is if we start to knock out repair factors that are involved in the Fanconi anemia pathway. So FANC-A, FANC-L. When we knock out these factors, we see a big reduction in the frequency of repair using these cross-linked templates. And so what we think this means is that the repair activity that we're seeing, it depends on the Fanconi anemia pathway. And so again, we're interested in these cross-linked templates because we think it can tell us which factors are acting specifically on the template. And so FANC-A, FANC-L, FANC-D2 that I talked about earlier on seem to be playing some role in the template molecule and altering double-strand break repair outcomes in that mechanism. The other interesting observation is if we inhibit a factor called ATR, we seem to lose this preferential HR using cross-linked templates. And so the bar plots in red on the left have the frequency of HR shown on the y-axis. And so uncross-linked template gives us maybe 5%. Cross-linked template gives us maybe 30%. If we repeat this experiment in the presence of ATR inhibitor, the frequency of HR using uncross-linked template doesn't seem to change. The frequency of HR using cross-linked template goes down dramatically. And so we think this is another mechanistic insight into how the decision to do end joining or HR is made. So the whole reason why we introduced these crosslinks in the first place was to do proteomic experiments, right? To identify proteins that are specifically bound to template molecules and play a role in the HR or end joining decision. And so we have actually done those proteomics experiments. So we have cross-linked templates on the left, we have normal templates on the right. And basically what we can do is we can look at factors that are enriched on the cross-linked templates relative to the uncross-linked templates. And so we do see Fanc-A, it is modestly enriched, but there are a whole lot of other factors over on the right side of this distribution which are much more robustly enriched. And so we think there's a lot of new biology here that we can start to take apart, right? We can understand these factors that are bound specifically to cross-linked templates. Okay, so to try to bring these stories back together, we think that there is an intermediate step, which I've termed quality control on this slide, where cells can simultaneously explore end joining versus homologous recombination strategies to repair a double-strand break. We think that Fanc-D2 specifically acts at this quality control step. And we also think that changing the abundance of template-specific repair factors, so these orange, these yellow ovals here, can dramatically alter the balance of end joining or homologous recombination outcomes. And finally, interstrand crosslinks in template DNA. While we don't have the mechanism fully worked out at this point, it can definitely boost homologous recombination, so it can sway the decision towards homologous recombination. And so we think this is really interesting for three reasons. First of all, it's just going to give us a better understanding of how DNA repair works, whether that's in biotechnology or in the context of DNA replication or just spontaneous DNA damage. We're really interested in the idea that understanding repair factors that act on template DNA will allow us to make gene editing reagents that are much more efficient. So, if one way to think about double-strand break repair, especially homologous recombination, is the cell has to do all of these steps in order to get from its starting point to final repaired outcome. And so if we understood the repair factors that were involved in all those steps, we could sort of start to do some of the work for the cells. So we could start to preload repair factors onto template molecules, maybe make it much more likely that the cell would then use those molecules as substrates for homologous recombination. And lastly, and this is a new area of research in the lab, we found that if you put interstrand crosslinks into template DNA, it makes it much more likely that those molecules will undergo homologous recombination. There's another famous agent that introduces interstrand crosslinks, and it's called cisplatin. It's given to about 25 million cancer patients each year. And one of the things that we would like to know is, are we changing the frequency of homologous recombination in patients that are treated with cisplatin, right? So if we're treating these patients with cisplatin, if we're introducing interstrand crosslinks in their genome, are we sort of, from an organism-wide perspective, altering the balance between end joining and homologous recombination for the repair of spontaneous DNA damage in those patients? And so this is sort of where we would like to go with one of these projects. I would like to thank the people that did all of the work that I talked about today. So Julian Bacall is a staff scientist and a lab manager in the lab. Hannah and Christine, I talked about a lot of their work today. They're graduate students. John Vu is joint between my lab and Brooke Gardner's lab and is working on a project that I didn't talk about today. And then the undergraduates, Carmen, Katie, Amy, and Amanda, contributed to the projects as well. So thanks very much, and I'm happy to answer any questions.
Great. So if anyone has a question, go ahead and unmute yourself and ask. I was really surprised by how many things there are that need repairing. That was quite a shock.
Yeah.
Are those numbers for cells that are actively replicating or all cells?
Yeah, those are all worked out for cells that are growing in dishes, and so they're actively replicating, and they're also being grown in a very high oxygen environment. So those numbers are probably lower, but I would say not like orders of magnitude lower for cells that are growing in more normal settings.
Mm-hmm.
We had a couple of questions.
Sure, go ahead.
Okay, the first question I asked in chat to you, Chris, but I now realize that it's not a real question, because I asked if you had to shut off end joining if you were looking at homologous, replication. So, okay, so obviously the cell makes the choice is what you're telling us. Okay, all right, so that's number one. Number two, in your schematics, you're using a circle versus the double strand.
Yeah.
Schematic. Are you using circular DNA? Is that what you're using, double-stranded circular DNA, or that's just a schematic?
So we use both. So we use circular double-stranded DNA and we use linear double-stranded DNA. And for almost everything, there's no difference between the two of them.
Okay.
Cell seems to be able to find the region within the circular DNA that it's interested in using. No problem. The big advantage of the circular molecules is they have this additional sequence that allows us to propagate them in bacteria, which doesn't match any human DNA sequence. And so, like, the way we see a lot of these reactions is we look at the sequence of the molecules involved. And so if we look in human cells and we find bacterial sequence, we know that we put it there.
Okay, thank you. I'm very impressed with this work. I wish I knew more about the field. To ask better.
I wish I knew more about how cisplatin works. So maybe you can tell me sometime.